is converted into tyrosine. So only one hydroxyl group is attached. So the name of the enzyme is Phenylalanine Hydroxylase. And rest of the reactions are same. So I am basically combining, I will basically covering only these two topics, and in general basic thrust will be on phenylalanine. Phenylalanine is an essential amino acid because tyrosine is not, because tyrosine can be produced from phenylalanine by our body. So phenylalanine has to be supplied in the diet. But by chance, if the phenylalanine hydroxylase is deficient in any individual, then this reaction will not occur. Do you understand? This reaction will not occur at all. And so phenylalanine will get accumulated. And that will cause problem that will be discussed today in detail. This is a question which is likely to be asked in your theory examination, maybe as a part of long question or maybe as a short note, and you may be asked in the viva. So I will discuss in detail about the problem if there is an excess phenylalanine consequent to deficiency of phenylalanine hydroxylase. Now, phenylalanine hydroxylase enzyme doesn't, function without anything. It requires Tetrahydrobiopterin as a cofactor. So that cofactor is also required. If that cofactor is deficient, then the enzyme will not function. So that is another problem. So that cofactor requires regeneration. So again, those enzymes which are causing regeneration of that cofactor, if they are absent, then also this will not be able to function. So we need a concerted effect and 100% presence of everything. Okay. So first I will show you the newborn screening will be a part of almost everything, and I have decided that I will spend a little more time on newborn screening. And although this class will be of almost 55 minutes duration, since on Wednesday from 3 to 4 p.m. There will be a gap, I will utilize that time. Maybe the class might extend even up to 4 o'clock. That you must be prepared, because next day there is a morning class. So you will get less time. So I will try to cover as much as possible, and remaining part I will cover on the Thursday morning, so that the time can be saved. And those which could not be covered, I am telling you at the earliest possible time, I will take 2 more SGD classes, so that in the SGD classes I will get 2 hours time. So in the 2 hours time, the entire syllabus what is there will be revised, and you also meanwhile read. And then you have an interaction. I think many things will be clear during that time. Anyway, the main thrust of today's class will be accumulation of high amount of phenylalanine and phenylketonuria. So phenylalanine is an aromatic amino acid. There are 3 aromatic amino acids: phenylalanine, tyrosine, tryptophan. And of course, histidine is there, that is having an amino group. it is having a both glucogenic and ketogenic amino acid. Why glucogenic and ketogenic? I am telling you because as I told you the carbon skeleton, I will show you once again that the carbon skeleton of all amino acids will be converted into 7 basically major metabolic fuel which will be channeled into different metabolic pathways. One will be acetoacetate, acetoacetyl-CoA, and acetoacetyl-CoA forms ketone body. Even from acetoacetyl-CoA you will get acetyl-CoA, and then that acetyl-CoA can also form fatty acids. So, from phenylalanine both fat, glucose can be produced. So, phenylalanine is converted to tyrosine, that is why it is called a sparing action of tyrosine. If adequate amount of phenylalanine is taken, there is no need to take tyrosine because it will be converted. So, this is called a sparing action of tyrosine on phenylalanine. So, predominant metabolism of phenylalanine, as I told you in the beginning, occurs through tyrosine only. So, tyrosine is a amino acid, it is incorporated in the protein. Phenylalanine is also an important amino acid, that is also incorporated in the protein. But the important products which are produced from phenylalanine are these, at least to name these things, and I will show you how these are produced. One is your epinephrine, norepinephrine, these are two adrenally metabolic hormone. Dopamine is basically a neurotransmitter. Thyroid hormones, iodination of tyrosine molecule is required to produce thyroid hormones. Melanin is synthesized in the melanocytes, these are present in the skin. Iris, the black color of the eye, what we see is basically due to the color of the iris and also the hair, because here we are having that pigment melanin. So, this is present in the skin, hair and eye. So, all these biologically important compounds will be produced. Now, if the phenylalanine is deficient in the sense that body cannot utilize phenylalanine because of the phenylalanine hydroxylase deficiency, all these materials will not be produced in adequate amount. But persons can live, for a longer life, but with a mentally retarded condition simply because tyrosine, if it is available in the diet, so tyrosine will start producing all these things. So, only issue will be that phenylalanine will not be incorporated in the protein. That will be a big problem because many proteins will not be synthesized properly. So, there is a codon, phenylalanine will be required for synthesis of protein. So, in the diet phenylalanine cannot be, I will explain you when I will discuss about phenylketonuria. So, these are the these are the biologically important compounds which are produced from phenylalanine, although these are produced through tyrosine only. So, you see this is a Delhi family. See, you look at the picture, all are white, skin absolutely white. Hair white, eye, if you look at the eye is also white. So, there is absolute deficiency of melanin synthesis, this synthesis. So, this is a family itself where both the parents, you see the father and mother, they are affected. They are affected means their children will be affected. I will tell you about the genetics of the disease. Now, if you look at the color of the eyes, the normal color will be like this, what we all have. This means this is the color of the iris. Now, you concentrate here, the color of the iris, can you see? there is no pigmentation. So somebody, some will have a different shades of color of the iris, maybe some will be hereditary, but they are not suffering from this type of disorder. I will tell you there are many varieties. So this is almost a normal, this is not also a normal color, but now you see here the iris color is almost absolutely white. So iris color of the black color of the iris performs good amount of function because it filters the light. Only light is allowed through this part, that is the lens. The black colored, absolutely black color in the center, and that is the place where through which the light passes and falls on the retina. So variety of colors will be there, and here you see the whole body, the whole skin is fully white. So that is called albinism. And this is why we got a case of, in the SMC OPD, you see this is patchy areas of white spots that you have seen. This is called vitiligo or leukoderma. The woman uses small tip that on the forehead. So because of the catechol, there might be reaction on the skin, and that causes a leukoderma. So leukoderma is one thing, vitiligo is another thing, and albinism is another thing. Albinism is complete deficiency. So I told you these are the metabolic fates of the carbon skeleton of the amino acids. Now you see phenylalanine and tyrosine enter, they can form fumarate, and once they can form fumarate, fumarate will enter into the TCA cycle, and from fumarate to oxaloacetate, and oxaloacetate can become a gluconeogenic precursor. Similarly, if you look at here, Phenylalanine-Tyrosine, it is producing acetyl-CoA. So this is also can form ketone body. So that's why phenylalanine is both glucogenic and ketogenic. Phenylalanine-Tyrosine both can act as a ketogenic and amogenic. So this is the catabolic pathway of phenylalanine and tyrosine 2. You have to remember this all the steps, because you don't know from where the question will come. Highlighting points here are the first reaction is an irreversible reaction, that is phenylalanine hydroxylase catalyzed reaction is irreversible. That means it is in one direction. I have deliberately written there are two reactions which are reversible. So note down these two reactions, the second reaction, third not, fourth not, fifth one is again a reversible reaction. Rest are almost all irreversible reaction, and here you see phenylalanine first getting converted to tyrosine and then to para-hydroxyphenylpyruvate, to homogentisate, to homogentic acid, to mallyl acetyl acetate, and through a process of isomerization, fumarate acetate, and then it breaks down into fumarate and acetoacetate, and acetoacetate gets converted into acetoacetyl-CoA. This is one pathway. So that you have to remember, this is available in the book. Don't try to write all these things, only write these two things. And you see the number of diseases. Why I have shown this part, this slide categorically. The first part is number one reaction, which I will be discussing in detail in today's class, is this reaction, that this causes phenylketonuria. Look at this picture, the last step here, that causes tyrosinemia 1. the reaction catalyzed by fumarate acetoacetate or fumarate acetoacetate hydrolase. There are two names that cause deficiency of this enzyme causes tyrosinemia. 1. Tyrosinemia. 2 is caused by tyrosine aminotransferase. This you note down. There is a high chance of committing mistake. The sequence is not same. 1, 2 and 3. 1 is here at the last reaction. 2 is here tyrosine aminotransferase or tyrosine transaminase. And third reaction is here para-hydroxyphenylpyruvate dioxygenase, or there will be some other name. This is called tyrosinemia 3. But at least forget about tyrosinemia 3, remember PKU, tyrosinemia 1, tyrosinemia 2. Don't get confused. Name of the enzyme here is fumarate acetoacetate or hydrolase. That is the last step. Okay. How norepinephrine and epinephrine and melanin are produced? You see here there is no norepinephrine, no epinephrine in this reaction. There is no epinephrine, but I told you there are several biological important components are produced. Now this is the reaction. Again you see PKU is there, tyrosinemia 2 is there, tyrosinemia 3 is there. But this is the reaction which produces this reaction catabolic pathway, but this is a separate reaction where DOPA is formed, norepinephrine is formed. This is the pathway for norepinephrine and epinephrine formation. Okay. Similarly for melanin, the pathway I will show you a simpler pathway, but this is a real pathway and this is a little bit complicated. Only thing is to highlight everything whatever is written in the schematic diagram are not that easy. So, eumelanin, whatever melanin normally we have as a black pigment, that is basically eumelanin and that is polymerized to form a black brown pigment melanin. There is another one which is produced also, that is called pheomelanin, that is polymerized to form red or yellow pigment. You have seen various shades of color in various individuals. So, this is because of the formation of this one. Those who are having a normal skin color, they are having eumelanin, they are having some altered color, they might be having a polymerization of pheomelanin. So, catabolic pathway is here, as I told you, that phenylalanine gets converted into tyrosine through a position at the para position, hydroxylase is there. Now, here tetrahydrobiopterin, write down, tetrahydrobiopterin is required as a cofactor. And here NADH is also required, oxygen is required. So, oxygen basically contributes to hydroxyl group and one atom of oxygen gets converted into water. This is an irreversible step. So, the phenylalanine conversion of phenylalanine to tyrosine requires phenylalanine hydroxylase, which needs tetrahydrobiopterin as a cofactor. Also it requires NADH and molecular oxygen. Molecular oxygen gets converted into, contributes to the hydroxyl group, and one atom of hydrogen is produced. Participates in the reaction of water. Now, this tetrahydrobiopterin, once it is used, that is called BH4 is the active form. So, during this conversion, this tetrahydrobiopterin is reduced to dihydrobiopterin. So, 2 hydrogen atoms have been removed. So, that needs to be regenerated. So, there is 3 enzymes are required. So, if 3 enzymes are also deficient, that means this regeneration will not take place, and that will give a similar picture just like phenylketonuria, but that will not be a classical phenylketonuria. It is not due to the deficiency of enzyme of phenylalanine hydroxylase, but due to its cofactor. So, a synergistic action and coordinated presence and functioning of all these things are required. So, here you see the hydrogen atom is shifted basically from this position to this position in tyrosine. So, this is called an NIH shift because this was discovered at NIH, National Institute of Health in USA. They have made a study with a radioisotope, and they found that during hydroxylation reaction, you see here hydrogen is there, hydrogen is there. Now, they have replaced it, hydrogen, the hydrogen has been replaced here. So, there is shifting of hydrogen because, and that is why this is called deuterium, they have used it. So, that is called an NIH shift because the study was done at National Institute of Health. It is basically a USA federal agency having 27 research institutes carrying out research work, and they are having a website. Unfortunately, my first paper which was available in 1991, I did not know about all these things. The first paper was uploaded in full in the NIH website, that is US National Library of website, which you can see right now in a mobile in a second, but I had to spend almost 2 to 3 hours time to get this article in 1991 from the National Informatics Center, which is the only place available for internet in India. So, active coenzyme, as I told you, active form of biopterin is tetrahydrobiopterin. So, tetrahydrobiopterin is oxidized to dihydrobiopterin, a quinoid form. So, tetrahydrobiopterin is then regenerated by NADPase dependent dihydrobiopterin reactors, as I told you. So, that requires a regeneration. So, phenylalanine hydroxylase is present in many tissues, but maximum amount is present in the liver. It is present in the brain, it is present in the kidney, it is present in the intestine, but maximum amount of phenylalanine hydroxylase is present in the liver. So, this is what is regeneration, how it is regenerated. You do not require to remember all these things, only to highlight the importance, how it is done. So, this is a simpler reaction to remember, this you will get in your textbook. So, phenylalanine is getting converted into tyrosine by simple hydroxylation, and tetrahydrobiopterin is getting converted to dihydrobiopterin, and then that is regenerated to tetrahydrobiopterin. So, I have shown in the previous slide, this reaction is not that simple, it is a little bit complicated and it requires 3 enzymes, okay, but these 3 enzymes have not been shown here. Now, this is now the first reaction what I have shown, now I am breaking it. So, phenylalanine is getting converted to tyrosine, then para-hydroxyphenylpyruvic acid, then dihydrohydroxypyruvic acid, this is called homogentic acid also. These 3 steps are there. Although this is written in this form, 2 reactions are reversible as I mentioned you in the first slide. So, this is also homogentic acid, and then mallyl acetic acid is there, and there is an isomerization of this molecule to form fumarate acetate, and then it breaks down into 2 parts, and this enters into the glucogenic pathway, this enters into the ketogenic pathway. So, it produces tyrosine. The tyrosine, when it is iodinated, it produces T3 and T4. This will be discussed in detail during your thyroid hormone synthesis, and if I take the class, I will explain in detail. For the time being, you remember that the iodine is incorporated into the tyrosine molecule, and iodine is incorporated in the tyrosine molecule, okay, to form thyroid hormones. I am just showing you how these specific structures are produced. Like melanin, how it is produced? The structure, as I told you, phenylalanine, neomelanin, these are complicated. The simplest structure is to remember, it is to produce from dopa to dopaquinone to indolquinone to melanin. Okay. Now, step 1 and step 2 is having the same enzyme. This is tyrosinase, and they are causing hydroxylation. They are causing hydroxylation only. Tyrosinase enzyme in both the steps, but tyrosinase and tyrosine hydroxylase are different. Both causes hydroxylation reaction. Now, you see the tyrosinase is present in melanoblast, means which produces melanin. That produces dopa, which produces melanin, whereas tyrosine hydroxylase is present in the adrenal medulla, which produces adrenaline and norepinephrine or noradrenaline and epinephrine. So, even if tyrosine is deficient, albinism, epinephrine synthesis will be normal. As I told you, we will go to the previous slide. See, here one part is here, norepinephrine, epinephrine and melanin synthesis is this one. This part is for melanin synthesis in detail. Now, melanin synthetic pathway is this one, and you see tyrosine and tyrosine hydroxylase, these are the two different enzymes. Now, clinical application of melanin, if you see the copper deficiency, again tyrosine is copper dependent enzyme. So, it is a metal enzyme. Now, if copper is deficient, the tyrosinase will not be able to function properly. So, all are interrelated. Malignant melanoma, if there is a melanoblast, if they function, overfunction, then they can cause a melanoma also. Leukoderma, as I told you, that is a localized depigmentation. Graying of hair is another issue. Albinism means whole body is white, there is no pigment. So, this is production of catecholamines, I told you, from tyrosine, dopa to dopamine to norepinephrine, from norepinephrine to epinephrine. This chart you will get in the book. So, you note down here, adrenal medulla contains pheochromocytes and neuroglial cells. In contrast, skin cell called melanoblasts. But here adrenal medulla produces adrenal hormones, and these adrenal hormones are produced by the pheochromocytes and neuroglial cells. So, if you go further, that norepinephrine to epinephrine and to metanephrine and finally to vanillomalonic VMA, vanillyl mandelic acid. So, when there is a tumor of the adrenal medulla, then this VMA excretion will be very much high. So, that is the importance. So, pheochromocytoma is a tumor of adrenal medulla, and when that becomes overactive, then excess amount of VMA will be produced. Similarly, for neuroblastoma, then norepinephrine will be produced in excess amount. So, coming to the main point of today's discussion is inborn error of metabolism, that is due to phenylketonuria. Earlier in the urea class also and glycine, I told you that if the concerned enzyme is deficient, then the precursor proximal to that block will be accumulated, and that level of that particular substrate will be high. That particular substrate may be channelized to some other biochemical pathways, and this is the classical example, phenylketonuria, how it is channelized to some other biomolecules, and these becomes clinically relevant and gives a clue to diagnosis. Now, if the enzyme is deficient, substrate is there. So, substrate, if the enzyme is absent here, so product cannot be produced. So, product will be deficient and the substrate will be in excess concentration because of the enzyme. So, phenylketonuria, write down some important point. These are available in the book. I am telling you, not concentrate here, listen only. Rest you read from the book, whatever is to be noted down, I'll instruct you. So, this is an autosomal, this is a first and foremost, this is the most common amino acid metabolism disorder. Although I will show you the incidence 1 in 20,000, in your lifetime you will see, if in a government hospital, you will see almost 50 to 100 deliveries per night, per day, 24 hours. But hardly will come across with one case of phenylketonuria. Although it is estimated to be 1 in 20,000, it is not that common, because you may get phenylketonuria, but those deliveries you have not conducted. Okay. So, it is out of these genetic disorders are indeed very much uncommon and if not rare. Occasionally you will find it, but if you are alert enough, then only you will be able to diagnose. Particularly in the baby, it is very difficult to diagnose. Those who are trying to opt for pediatrics, please be very much serious and sincere in understanding this thing. Because in a newborn, 2 to 3 years, 2 to 3 days, even 1 week, if you cannot diagnose the disease condition properly, a permanent damage will occur. So, this is autosomal recessive inheritance. You know what is the difference between what is the autosomal recessive disorder? You must know this thing, otherwise you will not have an implication. Autosomal recessive, I will tell you in a short while. Note down, it is autosomal recessive disorder and the phenylalanine hydroxylase deficiency is there. It is classically the patient should be mentally retarded because phenylalanine cannot be utilized properly by the brain. Excess phenylalanine will be available and that will cause disturbance of this neurotransmitter synthesis, particularly norepinephrine, epinephrine and serotonin synthesis will also be decreased because phenylalanine excess will be there and it will try to compete with the other things. So, because of the neurological damage, there will be neurological features and that will be diverse in nature. And these neurological features, since these are diverse, these are common in many diseases. You cannot pinpoint from the neurological disorder that this is a case of phenylketonuria. But for phenylketonuria, there is a specific smell, that is a mousy odor. So, the child will also have a hypopigmentation because of the decreased melanin synthesis. Okay. So, one thing is sure that if there is a block here, so phenylalanine will be accumulated in a high amount. So, that is called a hyperphenylalanemia and the gene is located for every enzyme. There is a location of the gene where it is located. It is in the long arm of chromosome 12 and there will be locus. You will be told all these things in your class also, in genetics class. Now, this you concentrate, try to understand here. This is the heart of this today's class. So, phenylalanine cannot get converted into tyrosine. So, phenylalanine will have this path. Phenylalanine excess will be there. So, excess phenylalanine, what it will do? It will try to find out some other channels where it can be produced into other products. So, one will be phenylpyruvate. In transaminase reaction, I told you, in transaminase reaction, everything gets concentrated into glutamate because glutamate dehydrogenase is the final common pathway. Here you see phenylalanine gets converted into phenylpyruvate. So, phenylalanine is an amino acid Now, pyruvate is a keto acid. If it is a keto acid is there, so one amino acid, one keto acid will be an acceptor of the amino group. So, alpha ketoglutarate is the amino acceptor and it will be converted to glutamate and this is a trans-amino acid based reaction. So, phenylpyruvate will be there because pyruvate is a keto acid. Pyruvate is not an amino acid, it is a keto acid. That is why its name is phenylketonuria. Phenyl group is there and ketone group is due to pyruvate and that is the reason it is called phenylketonuria. Okay. Now, this phenylketonuria will go further reduction because in your glycolysis you have seen the pyruvate to lactate conversion is there. So, there will be reduction of the uses of NADH. So, same thing will happen here. So, phenyllactate will be produced. Now, again there will be decarboxylation in this reaction that is called it will produce phenylacetate and this phenylacetate is responsible for giving, note down, phenylacetate is the chemical which is responsible for giving the typical mousy odor of the body of the urine, that is responsible, not the phenylpyruvate, neither phenyllactate. Phenylacetate is the main compound, maybe other products also, but again phenylacetate can conjugate with glutamine to form phenylacetyl glutamine. This reaction you try to copy in the examination hall. So, that will indicate your understanding about the phenylalanine and if you remember this thing you will be able to understand if you answer rest of the part. I am explaining it to you. So, this is available in the book, you just have a note, no need to note down. So, phenylalanine hydroxylase will be deficient and this pathway will not operate. So, conversion to phenylalanine to hydroxylase, again I told you it requires reduction and regeneration of tetrahydrobiopterin. So, if that pathway the enzymes are deficient, so phenylalanine will again get accumulated because it will have the same picture. So, 3 enzymes are there. So, because of the variation, the classical phenylketonuria will be due to deficiency of phenylalanine hydroxylase and rest 3 atypical hyperalanemia because of the deficiency of the tetrahydrobiopterin and these enzymes are responsible for regeneration of tetrahydrobiopterin. So, deficiency of these things will also cause hyperalanemia, but it is not the classical type of phenylketonuria. Okay. The altered metabolism will be there, tyrosine will not be produced, tetrahydrobiopterin cannot occur, dopa to dopamine will not be there. So, dopa decarboxylase produces, write down one thing. So, dopa to dopamine is produced by the dopa decarboxylase by decarboxylation reaction. So, this is a PLP dependent decarboxylation. So, there will be no dopamine. So, dopamine is a neurotransmitter. So, neurological deficit malfunction is due to dopamine. Okay. So, melanin biosynthesis will also be affected. So, there will be hypopigmentation. So, whenever there is a question, that is a child, baby 1 year old or 2 month old or 3 month old baby having neurological disorder, convulsion, seizure, along with hypopigmented patches and mousy odor. Mousy odor, if you see, if you smell mousy odor written somewhere, it is phenylketonuria. Newborn baby or a 1 year or 6 month old baby or a 1 year old baby, 2 year old baby, whatever it be there. Usually within 6 months it is having hypopigmented patch with seizures, convulsion, neurological deficit and mousy odor. Look at this thing whether mousy odor is there or not. So, mousy odor means it is phenylketonuria, it is due to phenylacetate, that you have to write down. Hypopigmented patches will be, you will get it because of the melanin conversion, because it is not albinism. Why not albinism? Because tyrosine you are getting in the diet from the protein. So, it will not cause a absolute deficiency of melanin synthesis, because tyrosinase enzyme is not deficient. So, there will be altered metabolism because tyrosine will not be converted to catecholamines. Why neurological deficit? Tryptophan will not be converted to serotonin. This is neurotransmitter. Catecholamines are serotonin and neurotransmitter. So, tyrosine synthesis, there is a block in phenylalanine hydroxylase. There will be tetrahydrobiopterin synthesis. There will be again catecholamine synthesis will be compromised. Serotonin biosynthesis will also be compromised, because what will happen in the central nervous system, the high amount of phenylketone will compete with tryptophan, and it will not allow tryptophan to enter. The serotonin production is made by the tryptophan. So, less amount of serotonin will be produced. That is the mechanism. So, phenylalanine is a large neutral amino acid. So, high amount of phenylalanine will compete, just like a competitive inhibition. You have read in the enzyme that if the amount of something is high, that will competitively inhibit something else. Here it will inhibit the competitively inhibit the transport of other amino acids, and because of that thing, the other proteins, other amino acids will not be able to enter into the system, because phenylalanine saturate the transporter system to a significant extent, and it will not allow the other amino acids, particularly tryptophan, to enter into the neuronal cell, and it will cause decreased serotonin synthesis. And these are the reasons why less dopamine, less norepinephrine, less serotonin. These are the causes for neurological symptoms of phenylketonuria. So, these things will accumulate. So, elevated phenylalanine will be there in the tissue, and phenylalanine will be degraded to phenyllactate, phenylacetate, phenylpyruvate. I have shown it to you. The symptom of phenylketonuria, as I told you, CNS symptom will be there, and it will be due to low dopamine, low norepinephrine, epinephrine, and low serotonin. This is the cause of, these are the causes of serotonin, and there will be a variety of neurological symptoms. This you will read from a book. Hypopigmentation will be there, patchy hypopigmentation, not the whole, because of the decreased synthesis of melanin. There will be fair hair, blue eyes, or albinism. Mousy or musty odor is a characteristic feature of phenylketonuria, and that is due to phenylacetate, not due to other things. So, phenylacetate gives the mousy odor, that you must remember. So, heredity. See, what is autosomal recessive? When both the parents are having the gene, only possessing the gene will not cause the disease, and the person will not know that he is having a disease. Now, during gamete formation, there is a chance that one gene will be having an affected gene, the other one will be having a normal gene. Now, during fertilization, there is a probability that by chance a normal gene from one individual will interact with the normal individual, it will give birth to a normal baby. Now, if a normal gene gives interact with a recessive gene, defective gene, that will cause a carrier formation what the parent is originally. Now, by chance, if both the carriers marry together and both the defective genes are produced in the zygote, then only the disease will occur in autosomal recessive disorder, otherwise not. So, there is a theoretical probability that only 25%, even if two carriers marry each other, there is a probability of 20% chance of having an affected child. 25% will be normal child and 50% will be carrier. That means 75% will be not be knowing that they are having a disease at all. Only 25% chance that if they have that disease, both the genes from accidentally both the parents, then there is a possibility of having the disease. Sir has come anyway. So, there is a propaganda of thalassemia carrier testing detection, you must be knowing the active camp is going on, and for that I had spent nearly 5 years time to study how much justified it is to have a thalassemia testing before marriage. Basically, I have written an article in 2006 in a local newspaper, because instead of doing that thing, if you start testing for thalassemia carrier, and if the person who is having a carrier status will be simply stigmatized, and there is a bleak chance of having a arranged marriage. He will not be married at all. Nobody is willing to willing to have want to marry that person. So unnecessary stigmatization before marriage I don't want, and that's why I have written in Bengali. In 2000 there is a popular demand that it needs to be translated into English. So I published an article also in English in our Durgapur Steel Plant internal journal. I can have an at-length discussion on this topic. Only thing is that since there is a chance of marrying a carrier is a chance. Chance of having by chance having a defective baby is another issue. So just to do that thing, unnecessarily stigmatization of a patient, of someone who is otherwise healthy at a school going age or during college life before marriage, will actually mentally shatter that individual and the family also. So that's why I am deadly against carrier testing for any genetic disorder which is having autosomal recessive in nature, because we don't know whom we are going to marry, whether a normal person or a recessive person, and what is the chance of having that affected baby. That chance, if it is there, then there are many chances. I can sustain an accident during year or even in the car, and I can die anywhere. So that was my point of logic. I will have a debate or discussion, whatever you say. I have many things to tell you, but please be prepared on Wednesday class. Even 50% class I could not cover in phenylketonuria. So this part will be covered and other parts. Okay. So this class might extend up to 4 o'clock. For that you must be prepared. So I am telling you because there are extensive campaign, but before you get yourself tested for thalassemia, be a little bit careful. I will discuss in detail. By chance if you are detected carrier, means gone. I am telling you in reality. Nobody is going to marry you, and you will be suffering from mental distress unnecessarily that I am having thalassemia carrier status.